The main real focus and function of HLA is to really help the body and its immune system distinguish self from non-self. So this is directly relevant in a cancer situation where you would want the immune system, and it would incorporate HLA in one of the mechanisms. You would want the immune system to eliminate these cancer cells. But unfortunately, cancer cells, they can be fiendishly clever, and many types of cancers have developed many different mechanisms by which they are able to actually evade the immune system.
Welcome to Science with a Twist, a podcast for curious people who enjoy exploring how science impacts our daily lives. From innovations in biopharma and diagnostics to technology that improves food safety and fuels advances in clean energy, it's all thanks to science. In each episode, members of Thermo Fisher Scientific's team talk to experts who are defining the next advances in science and technology that enable us to thrive. Let's get started.
Welcome to Science with a Twist. I'm your host, Terry Summers. Do you ever wonder what happens after you check the box on your driver's license indicating your willingness to be an organ donor? The process of organ donation is quite complex, involving various medical evaluations, diagnostics, and waitlist algorithms. But this process can differ in different parts of the world. However, at the core of it all is a microscopic set of proteins called human leukocyte antigens, or HLAs. HLAs revolutionized the science of transplantation back in the 1950s. HLAs are very useful biomarkers, which means they provide important information about the immune system. Essentially, they help doctors understand how a patient's body might accept or reject a donated organ. As our knowledge of the HLA system has evolved over the past 7 decades, so too has our understanding of its utility beyond transplantation. On today's episode, we're going to talk about these familiar, reliable biological tools, HLAs, and their growing uses in healthcare, particularly in cancer immunotherapy and autoimmune disease. Our guests today are Marcelo Fernandez-Vina, professor for the Department of Pathology at Stanford University Medical School and director of Histocompatibility and Immunogenetics Laboratory at the Stanford Blood Center. Marcelo has a PhD in internal medicine from the University of Buenos Aires Medical School, and he has worked in the fields of histocompatibility and immunogenetics for more than 3 decades. Prior to joining Stanford, he served as professor in the Department of Laboratory Medicine at the University of Texas MD Anderson Cancer Center, and has previously served as president of the American Society for Histocompatibility and Immunogenetics, as well as a member of the board of directors of the United Network for Organ Sharing. Also joining us is Dave Lowe, Senior Director of R&D for Transplant Diagnostics at Thermo Fisher Scientific. Dave holds a PhD in transplant immunology from the University of Warwick, and he previously served as the head of the Transplant Immunology Department for the Royal Liverpool and Broad Green University Hospital NHS Trust in Liverpool, England. Welcome, Dave and Marcelo. Thanks for joining us today.
Oh, thank you.
Thank you, Terry.
Marcelo, in the introduction, I mentioned that you're an expert in the fields of histocompatibility and immunogenetics. For non-scientists like me, could you please clarify what those two fields entail? And then can you tell us what sparked your interest in HLA?
So immunogenetics is defined as a discipline as well that studies the genetics of the immune response. The HLA system is the designation of a genetic system that we found in many species, and in humans we call it HLA. So what these genes are defining are proteins that play a role in tissue compatibility, as mentioned before, but also play a major role in determining the immune response of a given individual. It determines the responses to different pathogens and viruses, and we can discuss later about this. And indeed, that is a discipline that has been evolving over the years.
What sparked your interest in this field originally?
Yes, when I graduated in biochemistry, where I grew up, discipline that is so-called laboratory medicine, I wanted to really get into something more exciting than just doing the current diagnostic tests. And HLA really presented a great opportunity. It was transitioning from the early definition of the system to many, many new applications. And indeed, I thought that it was an opportunity of studying and performing research and developing some methodologies that would go beyond the common laboratory practices that I was trained to do.
Thanks for sharing that. Dave, how about you? How did your HLA journey begin?
Well, it's probably nowhere near as interesting as the tale Marcelo just told, but really for me, it all began back in 1998. So compared to Marcelo, I'm still a newcomer, and I graduated from university for the first time in 1998. And like most students, I'd studied incredibly hard and also enjoyed myself a little bit during that last 3 years. So I really needed a job. And that's when I saw an advertisement on a university notice board for a scientist role in the histocompatibility and immunogenetics laboratory for the National Blood Service in Birmingham, UK. I had no idea what histocompatibility and immunogenetics meant, but I did some quick fire revision and went for an interview and got the role. And then once I was in the role, as Marcelo will probably testify, once you start down this interesting path of HLA and you realize all the complexities and all the things that HLA does and how important it is and how crucial to successful transplant, you get hooked. And I've been in this world ever since.
So, Marcelo, what exactly is HLA and what is its role in the immune system?
HLA is defined as a genetic system that encodes for some proteins that play a central role in the immune response. The proteins are called this way because it's called, or the system is called human leukocyte antigens. And indeed, it shows a lot of genetic variants that are important in the determining compatibility for transplantation. And there are also these variants at the same time may play a role in a specific individual in defining the genetic profile for mounting a specific immune response.
And how does that translate into being a useful tool in transplantation?
Yes, by testing and determining these variants present in an individual, let's say in a patient, and the variants of HLA that are present in another individual, let's say a donor, one can assess the compatibility in HLA. In other words, we look at how similar or how different are these proteins in the patient and in the donor. And indeed, we can estimate the risk for rejection of an organ, or we can estimate the risk for example, in hematopoietic stem cell transplants, for a donor developing, the donor graft developing cells that will attack the tissues of the patient that we call graft versus host disease.
Thank you, Marcelo. That's really helpful. So, Dave, what is Thermo Fisher's role in the transplantation process?
Yeah, no, that's a great question. And I feel really fortunate because I'm in the position of being able to lead the R&D team in the Thermo Fisher's transplant diagnostics business. And for me, if I could just take a few moments just to talk about the history of that business and that company. So actually, the transplant diagnostics business was its origin came from a small spin-off company that was begun out of UCLA in 1984 by somebody Marcelo would know very well, Dr. Paul Terasaki, who described some very seminal work in the 1960s, which actually showed the importance of a pre-transplant serological cross-match and how this could be used to essentially make sure that your transplant was safe at that time. And again, this is one of those moments that we talk about in terms of just being a watershed moment that really drives the field forward. Well, anyway, Dr. Terasaki then went on in 1984 to found a company with 7 of his peers from UCLA, and that company was called One Lambda. And initially, that company was the first one to commercialize serological tests for HLA typing. And that business grew, and that business grew very successfully. It expanded beyond serological typing. It expanded into the new era of molecular, so DNA typing for HLA. And it also led the way in the study of HLA antibodies and designed revolutionary reagents, kits, assays to determine the status of patients who had antibodies to HLA. Because as Marcelo and I know very well, it's patients with large amounts of antibodies to HLA that really challenge us in the clinics, and we have to be creative sometimes in order to get those patients transplanted. So where we are today in 2025, we still offer a wide range of HLA typing assays. We still supply the real old school serological assays, but right through to the newest sequencing technologies. We provide many different options to suit every lab's needs. We supply antibody detection products, and we are really expanding into post-transplant monitoring as well. So we support all forms of transplantation, solid organ, bone marrow, stem cell as well. And we're even beginning to expand outside of transplantation. And I know we'll talk about this a bit later on, but we're looking now to support labs in the areas of companion diagnostics, biopharma, vaccine development. So the possibilities for HLA are almost limitless.
Thanks for sharing that. Really interesting. Marcelo, for decades now, HLA typing has been an incredibly useful and reliable tool for transplantation. But when did it begin to evolve beyond transplantation?
Well, two important things have happened and kept evolving. We started defining the HLA system better. For example, we started characterizing what we call the HLA class 2 variants, and that led to perform specific studies. And indeed, investigators learned how to conduct studies that would identify individuals that could be predisposed to development of some conditions. It was focused in the late 70s and early 80s in identifying if there were HLA variants that associated with susceptibility to some of these autoimmune disorders that are common. For example, juvenile insulin-dependent diabetes or IDDM, rheumatic conditions like ankylosing spondylitis, rheumatoid arthritis. And in addition to that, we started learning other associations or identifying other HLA associations that at the time were not thought, that involve diseases that were not thought to have an autoimmune basis, for example, narcolepsy. And indeed, this led to starting some mechanistic studies that could identify the causes of the disease. And indeed, it has been characterized that some cells that produce a specific hormone were destroyed and thought that was because of immune phenomena.
Oh, interesting. Dave, last year, Thermo Fisher announced that our C-Core HLA typing kit had received FDA clearance for a new treatment, specifically a T-cell therapy that boosts the body's immune system so that it can fight advanced synovial sarcoma, which is a rare, slow-growing cancer that occurs in soft tissue. What's the connection there?
Yeah, no, it's a great question. So again, referring back to something that Marcelo said earlier on, if we just recap that the main real focus and function of HLA is to really help the body and its immune system distinguish self from non-self. So this is directly relevant in a cancer situation where you would want the immune system, and it would incorporate HLA in one of the mechanisms, you would want the immune system to eliminate these cancer cells. But unfortunately, cancer cells, they can be fiendishly clever, and many types of cancers have developed many different mechanisms by which they are able to actually evade the immune system. So this is where some T cell therapies, so T cells are a crucial cell that is a cell type that is central to the immune system and is integral to our immune responses. So there's been a lot of work and focus on the area of developing T cell therapies. And these are engineered therapies which are designed to activate the immune system to fight back against the cancer. But in order to be able to do that and to activate that immune response, many of these therapies need to interact with specific HLA proteins. So if you can imagine a bit like a lock and key mechanism, a particular T cell therapy, if it can try to hone in on that cancer, but it must also fit the particular HLA protein found on that patient's cell in order to work. So it needs to have something it can latch onto, and that's where the HLA comes in this scenario. So what we offer with our C-Core HLA typing assay is a high-resolution HLA typing assay, which will enable you to identify which patients would benefit the most from these critical therapies.
So, Dave, how does the HLA typing come into play in the T cell therapy for advanced synovial sarcoma?
Well, the synovial sarcoma treatment that we're referencing is actually engineered from the body's own cells. So they take out the cells of the patient, they make some very clever engineered changes and then put them back in. So this is what we would call an autologous therapy. But conversely, some other T cell therapies that are available are actually derived from T cells that come from a third-party donor. And in this case, rather than being autologous, we call them, those are sort of allogeneic. So they're from a different genetic individual. So a bit like the example I described earlier, we need to know the HLA typing of these allogeneic therapies. We touched on the fact that there are many patients out there that have a lot of antibodies against HLA. So we want to make sure in these cases that any patient who is going to receive one of these allogeneic therapies doesn't already have antibodies directed against the HLA expressed on that therapy. Because if it does, it would cause an immune response, and actually the patient themselves would attack the therapy instead of allowing the therapy to attack the cancer.
Oh, that's very interesting. Thank you. Marcelo, in your research, are you finding that HLA plays a role in any other health conditions or treatments?
Yes, indeed. And going back to the application of next generation sequencing, it has allowed this methodology to really go into the detail of definitions of or distinction of these variants of HLA with much more accuracy. For example, some performing HLA typing may help us predict adverse reactions when we are going to administer a specific drug. And actually, this is what is called an area of pharmacogenomics. And for example, in one of the antiviral drugs, abacavir, used in HIV infection, shows often adverse reactions in individuals that have an allele or a variant of HLA, and specifically is called, the allele is HLA B5701. So physicians would take into account the HLA type of a patient before administering this drug. In a similar way, for example, in the administration of some antidepressant drugs, like let's say like carbamazepine, some individuals may develop an adverse reaction that actually involves systemic complications that may affect skin and other organs as well. And for example, for the alleles or the variants called B1502, B1511, associate with susceptibility to these adverse reactions. And indeed, doing this high-resolution testing allows us to really be more specific to determine what individuals carry the predisposing allele and what individuals do not. In recent work we have just published the adverse effects that of a drug that or some drugs, the antagonists or agonists of interleukin 1 and interleukin 6. These drugs are used in diseases where there is systemic inflammation, like COVID, like systemic arthritis. And when the drug is administered, there are some adverse events with a specific, when the patient has the specific alleles. For example, this would be the RB1 1501. We just published it, and indeed, these tests need to be performed before doing the administrations when there is a well-documented adverse effect when these drugs are done for susceptible individuals. So there is the requirement for performing HLA typing for a patient that is going to be receiving different types of drugs.
Dave, how might HLA research contribute to future treatments for immune-mediated disease?
So as Marcelo mentioned so eloquently, HLA testing can really help to identify the presence or the absence of specific HLA variants or alleles that have been linked to specific diseases or drug hypersensitivity reactions. I think one of the most relevant and the one that would resonate with many people is that there is a very clear linkage between HLA and rheumatoid arthritis. And in fact, it's just a very short sequence on the HLA molecule that is potentially predisposes us to rheumatoid arthritis. HLA is also playing a role in the design of effective cancer vaccines. And I know there are a number which are currently in clinical trials at various stages. But if I could, I'd just like to go just slightly outside of HLA and talk about some other genes that are found in a region of the human genome alongside HLA. And this region, which is found on chromosome 6, is called the major histocompatibility complex. And it's actually the most highly variable, or as we would say, polymorphic, has many forms. So it's one of the most it is the most polymorphic region of the entire human genome. And there are many other genes located in this region which are related to our immune response. And it reminds me of the example of an iron overload disorder called hemochromatosis. And for many, many years, we would test the HLA association in our laboratory for this disorder, and we would be looking for presence in the individual of the HLA-A3 protein. But actually, that was a red herring all along, and it was actually a gene that was found not too far away on chromosome 6 from the HLA-A gene, called the hemochromatosis or HFE gene. And this iron overload disorder actually wasn't caused or the HLA didn't play a role at all. It was actually 2 point mutations, so just a single amino acid change at 2 positions in the HFE gene, which could predispose the patient. So I think that's something that we can expect maybe in the future, that as we expand beyond HLA and we explore the variations in the MHC generally, we may find links to diseases and other sort of relevances to our immunological response.
What do you see for the future of HLA within the healthcare ecosystem?
Yeah, I'll dive in there. So again, I'm going to move outside of HLA, not for the first time. And I'm going to talk about something called SLA, which is swine leukocyte antigen. So really, we're now talking about pig organs. We're talking about xenotransplant. So I'm pretty sure that you've all seen on the news that over the past year or two, there's been quite a lot of attention and activity around the area of xenotransplant, particularly in a small number of large centers in the United States. They've been reporting on ever more success and being able to make patients survive longer and longer with transplants from organs derived from pigs. So I think that, I don't know whether it's going to be mainstream in 3 years, 5 years, but there's certainly a huge amount of effort going on in this area to develop this new source of organs. Because that's the real driver here, is that we don't have enough donor organs to go around. So if we were to empower ourselves with a new source that is essentially almost unlimited. I think the challenge again for people in the field is that xenotransplant will no doubt bring its own immunological challenges that we will have to consider. So I think there will be a need for diagnostics. There will be a lot of learning. So everything that we've spent our careers learning about HLA, maybe we have to relearn for SLA. But it's definitely very exciting.
Wow, interesting. Marcelo, what about you? What do you see coming down the road?
Well, I think we will follow a similar path in improving efficiency, in using smaller and being more sensitive, in having tests that are more accurate, and we can test more individuals. So as we were talking about, there is a number of scenarios that need to be controlled in specific when some research is being conducted. So really controlling for HLA variation is important. And so therefore, I anticipate that we do things in a simpler way. Our tests are simpler. Our tests are faster. The tests allow us for doing testing more individuals. Is what we're doing today better and faster with more resolution?
Great. That's a very promising note to end this discussion on. This has been a very informative discussion. Thank you, Dave and Marcelo, for taking the time to share your expertise with us today.
Thanks a lot, Terry. Really appreciate it.
Thanks for tuning in to this episode of Science with a Twist. This podcast is brought to you by Thermo Fisher Scientific, the world leader in serving science. If you enjoyed this episode, then follow Science with a Twist wherever you get your favorite podcasts. Or join us online at thermofisher.com/sciencewithatwist for all of the latest episodes.